southwest Germany, between the Palatine Forest and the Rhine, we find BASF's Agricultural Research Station. Here at Limburger Hof, research and development work is carried out on new fertilizers and crop protection agents. The development of Opus, the new cereal fungicide, began in these greenhouses and laboratories. Its active ingredient is epoxyconazole. It belongs to the triazole group of fungicides. The research and development work on the new product took 8 years and cost well over 100 million marks. Opus is effective against all important cereal pathogens. No other triazole combines so many advantages. 3 important properties make Opus a convincing product. Firstly, its broad spectrum of activity. It provides good to very good control of all important fungal pathogens of cereal crops. Secondly, the high level of effectiveness. Only 125 grams of the active ingredient are required to protect a hectare of cereals. Thirdly, the long duration of activity. The farmer can prevent diseases with fewer treatments. How does Opus achieve this? See for yourself. The search for new crop protection agents is expensive. Some 10,000 new test substances are screened each year in the greenhouses of the microbiology department. Perhaps they will include a winner, perhaps they won't. 10,000 tests. A special robot weighs out each new substance into a glass beaker. It mixes it with water and an emulsifier. Prepared in this way, the test substances are then sprayed uniformly on plants in special cabinets designed for this purpose. In the initial tests, BASF uses wheat, barley, rice, grapes and cucumbers, as well as the leaves and fruit of green pepper. The economically most significant diseases occur on these crops. In other words, pathogens diminish the yield or quality so seriously that it is worth doing something about it. After the spray droplets have dried down, the plants are inoculated with spores of the appropriate fungus. Here, wheat is being dusted with spores of powdery mildew. In the summer of 1985, BASF research workers discovered the excellent fungal activity of epoxyconazole, the active ingredient of Opus, for the first time. Epoxyconazole acts, for example, against the fungus that causes eye spot disease in wheat. The fungus grows at the base of the stem, from the outer epidermis, deep into the tissue. Infected plants are then unable to transport enough water or nutrients. Result? The stem rots, the wheat topples, there is lodging, and the yield drops. For comparison, healthy wheat. The plants can develop their full yield potential with Opus. Brown rust of wheat is spread by the wind and causes red-brown pustules on the leaves. The green leaf tissue surrounding the red pustules is destroyed. The leaves turn yellow. No further photosynthesis takes place. Crop losses are therefore considerable. The white spores of wheat powdery mildew are also spread by the wind. This cereal disease occurs very early, sometimes even in autumn. Wheat powdery mildew also reduces the green leaf area required for photosynthesis. Net blotch disease of barley has similar consequences. The fungus destroys the green leaf tissue. Here too, there is a drop in crop yield. Opus protects cereals from the stem and leaf diseases that have been shown. Opus also prevents the ears from being attacked by septoria. Protection, therefore, from head to foot. Cereals can fully develop their yield potential. Opus has good residual systemic activity. This means that Opus forms a fungicide depot on the leaves and stems. The plant takes up the active ingredient from there and transports it with its vascular tissue. In this way it gets everywhere, into the stem, into the leaves, and into the ears. Even new parts of the plant which grow after treatment are protected. The excellent fungal activity of Opus is both protective and curative. Why does Opus work so well? This is a question for my colleague Dr. Gold. He and his team are on the track of the different modes of action of fungicides. We now know more about the development of pathogenic fungi than we used to. We are more familiar with their strategies and their weak spots. We're therefore able to develop fungicides such as Opus that interfere with the pathogen's biochemistry with a high degree of precision, and so keep them in check. We know very well how our crop protection agents act. Opus protects cereals in 4 ways, both on and in the leaves. The first question is, how does Opus prevent the pathogen from penetrating the leaf? The surface of a leaf is viewed with a scanning electron microscope. The elongated epidermal cells can be clearly recognized. Their function is similar to that of the human skin. They seal the plant off from the environment. The only natural openings are the leaf stomata. Rust fungi grow specifically toward these weak points and enter the plants through these gates. The spores are carried in by the wind and germinate on the leaves. They form a blown-up cell precisely over the stomata, the so-called aprosorium. The aprosorium attaches itself firmly and forms a hypha that penetrates the epidermis. How does the fungus recognize the entrance gates? It recognizes them by their shape. This shape can be imitated in the laboratory in a model system. The spore then germinates on a prepared plastic film, just as on the surface of a real leaf. It forms the aprosorium on the elevated portion of the replica, because it thinks that this ridge is the stoma. It may be seen under the microscope and on the monitor how the fungus grows towards this ridge. The ridges now appear on the prepared plastic film like the grooves on a record. The film is cut into the right size, and the pieces are positioned for the subsequent transfer of the fungal spores. The rust spores are transferred from bean leaves to the pieces of plastic with a brush. In nature, the spores are spread by the wind. The film is turned over so that the spores are underneath. When it is mounted on a glass slide, a growth chamber is created. The spores will develop in these small chambers as if they were on a plant. They're provided with water so that they can germinate. If no fungicide is added, then this untreated chamber serves as a control.